The next presentation we have is, and also an expert in SPMs, Dr. Dalli, who's a professor of pharmacology at the Queen Mary College in London, which is also affiliated with Bart's Hospital. As I say, he's a true expert in the SPM area, has co-authored more than 150 publications in peer-reviewed journals, and he also has received a number of awards, including the Young Investigator Award from the Icosapent Research Foundation. And with that, please join me in welcoming Dr. Dalli to give his presentation. Thank you.
So good morning, ladies and gentlemen. Thank you for the kind introduction, and also thank you for welcoming me here. This is not usually the audience that I'm used to addressing, especially after a few days and a bit of a jet lag. So please bear with me if I sort of steer away into deep science. Every now and again I'll try and refrain myself as much as possible. This is just a little bit of a snapshot of the UK, and that sometimes we do have sun. It's not always there, but it does appear every now and again. And actually this is our institution. So there's a few disclosures that I want to bring to your attention, in particular the fact that I'm a founder in a company that is aimed to bring some of our basic observations into the clinic. And I should note that a lot of the work that I'll be presenting today was not done by myself, but my team. As you can see, we recruit very early on. We believe that you need to start training early on, as well as an extensive group of wonderful collaborators. And I'll mention some of these as I go along. And of course, the funders. So you're all very familiar with the diversity of fatty acids. There's all sorts of structures. They come in all sorts of shapes, sizes, etc. And they have a variety of biological functions, from maintenance of membrane integrity, to energy, to signaling molecules. And this particular aspect, the signaling molecule, is the one that we're primarily interested in. And before diving into this, I would like to just bring to your attention another key definition to some extent, and in particular this dichotomy between mediators and metabolites. So you're all very familiar with DNA, we're all familiar with proteins, we're familiar with enzymes, and enzymes by definition produce metabolites. Now these metabolites can either be bioactive or have either no known bioactivity or are inactive. And this distinction is really important in what we're going to be talking about, because the essential fatty acids can be converted either via enzymatic mechanisms or non-enzymatic mechanisms to a variety of metabolites that may be bioactive or not bioactive, and our interest is in these bioactive molecules, in particular molecules such as the resolvins, which you've heard about, prostaglandins and leukotrienes. And this eye chart here is really just to give you an idea of the work that's being done and the diversity of these molecules within biological systems. So each of the essential fatty acids that are depicted here gives rise to distinct families of these specialized pro-resolving mediators. They're grouped into four main families, the resolvins, the protectins, and the merisins, which are depicted over here. And a key aspect here is that the structures are distinct. And this point of structure I'll come to a little bit later in my presentation, because it's fundamental in understanding the biology. Our understanding of where the substrate for the formation of these molecules comes from is still not very clear. There are several studies that demonstrate that the essential fatty acids can either come from free fatty acid sources within, for example, peripheral blood, that during inflammation seeps into the site and feeds these biosynthetic pathways. But there are also sources within the esterified pools, within membranes, for example, that are released by, for example, by different phospholipases. Here is one example for CPLA2, for example, which in the microglia releases DHA to produce protectin D1, whereas in other settings, such as in the lymph nodes, this other phospholipase can release docosahexaenoic acid to produce resolvins and protectants that then are involved in reducing skin inflammation. So likely the mechanisms that feed the pathways will be different in different tissues and in different cell types. And this is an aspect that we're very interested in understanding better, and actually it's an active aspect of research. So this is the point that I was making earlier, this idea, this aspect of three-dimensionality, the structure aspect. As you appreciate in life and in everything that we have around us, there's defined structures, and these defined structures for the SPMs confer their biology through a lock and key mechanism. This is because each of these molecules activates cognate receptors. So the biology of SPMs is via the activation of cognate receptors, of specific receptors that recognize the distinct structures, and through this, a unique biology is conferred. A unique biology is activated through the activation of downstream signaling cascades. And so, although there's this wide array of structures, this wide array of biology, there is limited redundancy in that each of these receptors has a distinct expression profile on different cells and couples with unique signaling pathways downstream. And this is where the biology comes from. And here is a list of the known receptors for these SPMs, along with some of the ligands. And the biology for these receptors has been established using a diversity of approaches, for example, using transgenic mice that overexpress these receptors, or knockout mice that lack these receptors. And there is increasing clinical evidence for example, the importance of single nucleotide polymorphisms that lead to receptors that are either signaling deficient, so even though the ligand, the mediator, binds the receptor, it can't convey the downstream signaling pathways or inhibit the expression of these receptors on cells, and those links with disease. The biology of SPMs is now, at least in experimental systems, has been extended to many chronic inflammatory settings, at least from a pharmacological perspective, extending all the way from vascular disease, such as arthritis, heart failure, airway inflammation, diabetes, gastrointestinal inflammation. And as you can see, many of these are also the activities of the essential fatty acids that have been described over the very many years. And we also now know that the SPMs are produced in humans in various organs and tissues, both in the presence of inflammation, so for example in synovial fluids, in adipose tissue and in lymph nodes, but also in settings where there is no clinical inflammation, such as, for example, in plasma. And I'll come to some of the work that we've done recently on this. So this suggests that the biology of SPMs extends beyond limiting inflammation. It potentially is involved in also maintaining homeostasis, so allowing the body to stay healthy. And again, this is another aspect where the supplementation and the biology of essential fatty acids links in with what we're now starting to learn about the SPMs. We also know that the SPMs play key roles in not only regulating the inflammatory ability of cells, but also in conferring protective activities. So for example, we recently found that these molecules are involved in facilitating the differentiation of monocytes to macrophages, and endowing these macrophages with protective activities, with tissue reparative activities, as well as in the differentiation of T cells, which are part of the innate adaptive immune system, and in particular in this case in the differentiation of T regulatory cells, which are involved in the regulation of inflammation. So the biology is not simply that of dampening inflammation, but also of instructing the immune response how to better respond to an inflammatory signal. And we've recently become interested in trying to understand the physiology, and as you all are very aware, one of the key physiological mechanisms that our body is governed by are circadian rhythms, light, dark, day and night. And so we question whether under these circadian conditions we see also regulation of SPM levels. And indeed what we found was that in peripheral blood of healthy people, these are actually colleagues in the lab, and there was a diurnal regulation of the expression of enzymes involved. So these 15-lipoxygenase and 5-lipoxygenase enzymes are involved in the biosynthesis of this family of SPMs from N3-docosapentaenoic acid. And through this diurnal regulation of both enzyme activity, this neurotransmitter acetylcholine, there was a diurnal upregulation of the formation of these SPMs. And this was important in regulating the physiological activation of peripheral blood cells. So when we wake up in the morning, or just before we're about to wake up in the morning, as the heart rate starts to increase in preparation of getting out of bed, there is a physiological activation of peripheral blood through this increase in shear stress that happens in the vasculature. And actually this, in many conditions, can lead to stroke. And most of the strokes happen in the early morning time. And usually they are the ones that with most catastrophic clinical conditions. So what we found was that these SPMs in healthy people help limit this aggregation of white blood cells and platelets in peripheral blood, and to limit the potential for stroke. And in patients with cardiovascular disease, we observed that there was an increase in this adenosine that blocks the formation of SPMs, and this led to an increase in leukocyte and platelet activation. So these molecules are now going beyond, as I was mentioning, beyond limiting inflammation to actually preventing inflammation through dampening leukocyte biology as part of a physiological mechanism. So during the next few minutes, what I'd like to share with you is how we're thinking about supplementation and the role of supplementation in regulating and conferring some of this biology in different conditions. So with one of our colleagues, David Collier, who's an expert clinical trialist, we set up a very simple clinical study where we recruited healthy volunteers, and we used a randomized, double-blind, placebo-controlled crossover study design in order to mitigate for many of the different factors, such as interpersonal variation in uptake, in metabolism, in conversion, as well as any crossover between one dose and a different dose, because we wanted to really see whether there was a relationship, and Phil nicely showed some of this in some of the studies that they did, but we wanted to see whether in this particular case there was a relationship between supplementation and SPM formation in peripheral blood. And we wanted to avoid any potential confounders, including things like carryover between one dose and the next dose. So this was a study design. I'll be happy to go through it if you guys want any more details. This is some of the cellular and molecular work that we did, where we obtained peripheral blood sequentially from the same patient, from the same volunteer, either before supplementation or after supplementation, up to 24 hours. And in these samples, we simultaneously evaluated cell activation, cell function, as well as lipid mediator levels. And at the 24-hour interval, we also did RNA sequencing of the peripheral blood cells. And in order to look at the flux down these bioactive pathways, these are very rapid biosynthetic mechanisms. So the formation of lipid mediators from the substrate through the bioactive mediator through the end product can be extremely rapid. So we wanted to get a snapshot of these dynamic fluxes, so we simultaneously measure the substrate, the precursors, the bioactive mediators, some of the pathway markers, and some of the further metabolites. And using this approach, what we observed was when we supplement healthy volunteers, there was a dose-dependent increase in peripheral blood SPM concentrations. And this occurred very rapidly after supplementation. So the supplement was a single dose, single bolus supplementation. So within 2 hours, we already reached more or less the peak levels at the 3 and the 4.5 gram doses. And interestingly, we did not really observe much of a change in the classic icosanoids. So this really suggests that there is a preferential shunting of the substrate into the SPM pathways, even though in some of these supplements we do see that there is arachidonic acid. So the conversion goes preferentially into SPMs. When we look at function, and if you remember earlier I mentioned that we observed that there are diurnal regulation in peripheral blood cell responses, so we wanted to see whether this was in any way impinged upon by supplementation. What we observed was that in monocytes, and this was true for other cells, we see that there was a significant downregulation in markers of activation in these cells that was dose dependent from the supplement that was given. So this suggests that there was a relationship between supplementation and peripheral blood cell responses, and this is true for neutrophils, as you can see over here. And we were very intrigued by this observation that most of the effects that we see occur after 24 hours. So this appeared to be offset from the increase in SPMs that we see in peripheral blood. If you remember, the increase occurred primarily at around 2 hours, whereas the function seems to occur at around 24 hours. And same thing we see in platelets. So we wanted to dig in a little bit further to understand what's happening, and we conducted correlation analysis to identify specific molecules that correlate with specific changes that we observed in peripheral blood, and each of these dots links the molecule with a change in function, and we observed that there was significant correlations between specific SPMs and changes in activation status in peripheral blood cells. But because we observed that a lot of the biological activities that were conferred by supplementation occurred at a 24-hour interval, we did this RNA sequencing because it suggested that there was something beyond simply changing of function. And indeed, what we observed was that you actually change the transcriptome of peripheral blood cells. So you're actually not just simply changing the function in short term, but the function is changed at a long term, because the cells now have a different playbook, if you will, because the genes they're carrying are different. And when we look at what these genes that are differentially regulated do, we observe that there are aspects of immune regulation, metabolism of proteins, metabolism for energy, as well as some of the genes that are associated with diseases. So this suggests that with a single dose, through the rapid upregulation of SPMs, you can get reprogramming of peripheral blood cells that is long-lasting. And this is in line with what we know about SPM biology from experimental systems. So this is our current model, where if you have a supplement that contains the precursors for these mediators, they're converted in humans to SPMs, and these regulate different aspects of the white blood cell responses, including activation, function through phagocytosis. I didn't show this data, but we see that these molecules increase the ability of white blood cells to phagocytose bacteria, as well as they reprogram the transcriptome. So we wanted to see with a colleague of ours whether this translated to patients, and with Mike Canty at UCSF, we recruited patients with peripheral artery disease, and we used a similar paradigm where there was a dose escalation of the SPMs. We measured these in peripheral blood, and we observed that there was a dose-dependent regulation of SPMs in peripheral blood with patients with peripheral artery disease. And this linked with a downregulation of a number of markers that are linked with activation on circulating monocytes from these patients. And when we conducted correlation analysis between the expression of these activation markers and SPM levels, we found that there were significant negative correlations between peripheral blood SPM levels in patients with peripheral artery disease and the expression of activation markers on these cells. So one question that we've become also interested in understanding is, are all the supplements equal? So, you know, as you all very well know, there is a wide range of sources, there's a wide range of formulations. Do they all do the same thing? So with one of my postdocs, Agua, we profiled a number of commercially available oils, and what we found by measuring simply the precursors and the substrates for SPM formation was that each of these had a distinct profile. So the levels of these molecules were very different in each one of these substrates. So in order to really get to whether each one did something similar or different, we used a bit of a pharmacology approach. And so what we did was, we summed up the levels of these molecules in each of these products, and we did a dose-response analysis to evaluate whether if we incubate cells, human cells, with an equal amount of these molecules, do all the products do exactly the same thing? I should note, we also profiled some, the first thing that we looked at was at the ability of these products to upregulate SPMs, and what we observed by using the same amount of substrate, by normalizing for the same amount of substrate, each one of the products using the same cells from the same volunteers gave a different profile. So the ability to convert to SPMs changes based on the formulation. And when we look at function, this was also true. So this is a dose-response relationship on the ability of each one of these products to upregulate to inhibit the ability of macrophages to uptake oxidized LDL, which you may know is involved in disease progression in cardiovascular disease, and it's involved in foam cell formation. And you can see that each one of these products has a different profile, even though the levels of these precursors is the same. And this was also true when we look at the ability of these products to upregulate phagocytosis of bacteria by macrophages. And importantly, any of these biological activities were, as far as we can tell, were conferred through the formation of SPMs, simply because if we block, using a pharmacological inhibitor, SPM formation, this is an inhibitor that blocks 15-lipoxygenase, one of the main enzymes involved in SPM formation, you reverse all these protective activities. So really, even though the substrate is there and we normalize for substrate amount, not all the products have the same activity. And this suggests that other things within the formulation have a biology that can interfere with SPM biology, and this matrix needs to be taken into account as products are being developed. So with that, I'd like to leave you with some thoughts. So we know that omega-3 supplements upregulate SPM formation and reprogram blood leukocytes from healthy volunteers and patients with peripheral artery disease. Conversion of SPM precursors to bioactive mediators is supplement dependent and is linked on the ability of distinct supplements to regulate immune response. And supplementation may not be applicable to all diseases. This is a key aspect, maybe especially in those settings where you have inefficient conversion of substrates through, for example, as we observed in cardiovascular disease patients, because the enzymes may not be present, and one may need to look at potentially circumventing this through supplementation with or addition of monohydroxy fatty acids to jump the defective steps. So with that, I'd like to leave you with the take-home message that upregulating endogenous SPM formation or using SPM analogs and mimetics represents new pharmacological approaches in treating uncontrolled inflammation. And with that, I'd like to thank you for your attention.